Um, Ladies and gentlemen, you're looking tired. I will be really fast. Don't worry about time. Okay, I'm very glad to see you here. And today I would like to focus on the role of the human microbiome. In my opinion, often undervalued. In the development and manifestation of disease with an autoimmunity immune component. Viral myocarditis is our model for today. At first, what is the microbiome? In my opinion, microbiome is a dynamic collection of genetic material of all microbes which cooperate with human organism. I want to emphasize dynamic. It's a crucial feature of microbiome. Dynamism makes microbiome flexible and changeable. Plus, the human microbiome colonizes different epitopes, skin, mouth, gut, and even the single cell. And the factor of dynamism in various epitopes are different. It's different. The content of microbial genes in an organism is truly incredible. Their number is hundreds of times greater than the number of human genes, and each gene has a function in several functions, and we know about video hundreds of functions. In this case, absolute exact sequencing is impossible. Each cell of the human body, with a few exceptions, has a mutation. However, sequencing of selected cell group makes differences between the cells are leveled, and such small mutations do not affect the function of the organism. Thus, the human genome obtained from the parents normally remains stable through life. When we're talking about the human microbiome, the situation totally changing. Its incredible flexibility and agility allowed to adapt to a different condition and to cooperate with human body, performing vital function and making the human body superorganism. Just before a week of my speech, I was in hospital in Mayo Clinic, in Department of Genetic Diagnosis. The main task now is to identify viral genes, most they are working with human papillomavirus, embedded in human DNA, and the effect on the expression of different genes of human, respectively, the possibility of variety of disorders. In definite circumstances, some bacteria and viruses can enter cells. Some bacteria even form colonies within cell. In such cases, the interaction between the human genome and the microbiome of the epitope, primarily I'm talking about cell, is unavoidable. And as far as microbiome is dynamic and therefore more adaptive microbes, the result is obvious. The microbes change cell metabolism, cell cycle, and use cell for their own purposes. It does not seem so be something strange, but how can we, with this understanding, how can we protect our genes? The answer is very simple, by our immune system. We have different definition of immune system function. I prefer this one, that the task of the immune system is the maintenance of genetic homeostasis. Even the immune system, which is able to change, often cannot cope with the performance of their function when the microbes begins to work as a unit. Myocarditis is an inflammatory disease of heart muscle. It is an important cause of delayed cardiomyopathy worldwide. Viral infection is also an important cause of myocarditis, and the spectrum of viruses known to cause myocarditis has changed in the past 2 decades. The virus may get inside the cells. However, the interaction with the DAF complement decay accelerating factor is used as a receptor by some Coxsackie virus and different enteroviruses, is launching a series of pathological changes that lead to the reorganization of the actin and the possibility of the virus entering from the tight junction. After virus penetration into the cell, the viral proteases destroy the necessary cardiomyocyte-specific proteins, runs cyclin D1 proteasomal degradation, starts long and complicated apoptosis pathway. Here is the simplified. Triggers apoptosis of the cell. Thus, after penetration of the virus into the cell, function of cardiomyocyte is impaired protein synthesis, delayed cell cycle, apoptosis occurs. Destruction of cardiomyocyte virus causes the appearance of cardiac antigens, which after processing and presenting activate the range of the T-cell signaling pathway, which ultimately leads to the development of immune-associated destruction of cardiomyocytes and manifestation of the autoimmune process. Interaction between immune system cells and bacteria, virus, which have demonstrated by Mr. or Marshall, makes situation worse. Autoimmune process weakens the body's physiological function that opens the door for more and more new infection. I think that the system approach is the best way to control and prevent multifactorial disease development. What do I mean when I say system approach? Now, here we are speaking about biomarkers of different levels: genome level, RNA level, proteome level, metabolome level. But the big part of biomarkers are potential targets. Thus, we can influence on each level, but limited set of targets and have an exact but soft treatment effect. But only creating and clear understanding which bioinformatical instruments, of course, follow pathologic pathway maps enable to find biomarkers of null generation, determine targets, and discover high efficient drug based on personalized features. Thank you very much.
Thank you.